this mini exam video, I'll illustrate and describe the process of transcription in prokaryotes. Transcription is the process in which a sequence of DNA nucleotides is copied into a newly synthesized molecule of RNA, or in the most basic sense, transcription can be defined as RNA synthesis. Transcription is an essential process that helps us use the information in genes of our DNA to make proteins. There are three main stages of transcription: initiation first, then elongation, and finally termination. During initiation of transcription, the core enzyme of the sigma factor will bind to create a hollow enzyme called RNA polymerase, which I have drawn out above the DNA double strand. This is important because this is an enzyme that is responsible for unwinding the DNA double helix so that RNA can access the genes that need to be transcribed. With the help of the sigma factor, the RNA polymerase is able to bind to the region necessary for transcription, which is the promoter region. The promoter region includes the -35 control region, the -10 control region, which is also called the tau box, and the +1 site, which is usually the initiation site where the first 5' RNA nucleotide is transcribed. It should also be known that nucleotides coming before the +1 site are given negative numbers and considered to be upstream, while those coming after the +1 site are positive and downstream. In the diagram of initiation, the hollow enzyme, which still includes the sigma factor, has binded to the promoter region and caused the unwinding and opening of the double helix, and this opening up is called the transcription bubble. The opening up of the DNA occurs around the -10 process of elongation. By now, the RNA polymerase has released the sigma factor as it has already helped the hollow enzyme bind and is no longer necessary. The RNA polymerase goes along the DNA template strand in the 3' to 5' direction, so basically for every nucleotide on the template strand, the RNA polymerase adds a complementary RNA nucleotide to the 3' end of the RNA strand, which has accessed the DNA through the transcription bubble. Keep in mind the complementation of each nucleotide will be the same on the RNA strand it is for the coding strand, with the exception of thymine, which RNA will code as uracil as opposed to adenine. Come to the final stage of transcription, which is termination. The RNA polymerase will transcribe the DNA strand into an RNA strand until it gets the signal to stop from the sequence of DNA known as the terminator. Prokaryotes have two major strategies of termination, which are either row-dependent or row-independent. In this diagram, I have drawn out the row-dependent termination strategy. In row-dependent termination, the protein called Rho factor will bind to a binding region of the RNA strand, where it will climb up the transcript all the way towards the RNA polymerase, where it will then pull the RNA transcript and the DNA template strand apart through helicase activity, which will end transcription and the RNA transcript will break off. In this diagram, I've illustrated the row-independent strategy of termination. Row-independent termination occurs with specific sequences on the DNA template strand that are rich in C and G nucleotides, which causes the RNA transcript to fold in on itself and the complementary C and G nucleotides to bind together, which leads to the formation of a stable hairpin loop that causes the RNA polymerase to pause transcription. The formation of the hairpin is followed by the complementary UA region of the RNA transcript, which is a weak interaction with the DNA template strand. This weak interaction between the RNA transcript and the template strand, along with the paused RNA polymerase, enables the enzyme to fall off and the new RNA transcript is liberated. Now that transcription is ended, the new RNA transcript is ready to be immediately used for translation. In my final diagram, I've illustrated the occurrence of an insertion of a 5 base pair between the -35 and -10 region of a gene that codes for an enzyme required for production of the amino acid phenylalanine. If this insertion were to occur between the -35 and -10 regions, a mutation of the promoter region would result. If the promoter region had a mutation, the sigma factor may not be able to recognize the promoter region, and if the sigma factor cannot recognize this region, then RNA polymerase would not bind to the DNA and no transcription will occur here. Ultimately, the amino acid phenylalanine would not be made if the sigma factor is unable to recognize the promoter region, and the organism may have serious effects if this occurrence were to occur.